All right, let's look at the filtration process then. Now, your book actually divides all this urine formation up into three processes: filtration, reabsorption and secretion, and then water conservation. I'm going to kind of stick to that, but not exactly. So here's the renal corpuscle. We have our afferent and efferent arterioles, our glomerulus. As I mentioned, the glomerulus is a semi-permeable membrane. The capillaries themselves are fenestrated. And I mentioned that before when we talked in chapter 20 about the different types of capillaries. You have continuous capillaries, which is what we find in the brain and skeletal muscles and in the heart, pretty much most of the body. We have the fenestrated capillaries, which are found primarily in the glomerulus, and then we have the sinusoids, which are found in the liver and the spleen and a lot of endocrine organs. They're very, very large capillaries with large spaces between the cells that allow substances to move in and out pretty easily. So these are fenestrated. The other thing that we find associated with this glomerulus is that there is a group of cells called podocytes which actually surround and cover the glomerulus.
Do they surround the individual capillaries, or are they around the whole glomerulus?
They're around the whole glomerulus, and what happens is these podocytes, if this is the cell body, they send out these feet. In a way, they're very, I guess, structurally similar to astrocytes. They send out these feet, kind of like an octopus would, but then what happens is they send out these secondary processes. And what happens is that these processes create these little interdigitating folds. Let me just kind of bend this one around here. So you have this small space, very small space, between these secondary processes of these podocytes. So this is actually a secondary or a tertiary filtration barrier. In fact, let me go to the next slide. So here is the simple squamous epithelium of the capillary. You can see the fenestrations, and so substances will actually move through those fenestrations. They'll be forced through there due to that hydrostatic pressure. Now there is going to be a basement membrane here that acts as a slight barrier, but not much. And then we have, here's the cell body of the podocyte, here's that primary process, and here's the secondary process. You can see here how they kind of interdigitate like that. That adds a third layer, a third barrier to filtration. And so what you're going to get is you're going to get small substances kind of squeaking out and leaking out between those, in those little gaps created by those secondary pedicels. Let me show you this in the book. This is a cartoon of that, kind of a cutaway. So here's the blood. Here is the fenestrations. This is the endothelium of the capillaries. Here's the basement membrane. And then here is those foot processes, those pedicels that are kind of interdigitated. And there are these little tiny gaps. So substances that are fairly large or of a certain diameter are able to squeeze through those fenestrations. But then a lot of things are turned back because they can't fit through those interdigitations. So that's why the glomerulus is a semi-permeable membrane. It doesn't select what goes through like a cell membrane. Things go through based on size. And this shows us that, blood cells, large plasma proteins, large negatively charged molecules, and substances that are attached to transport proteins in the blood do not or should not actually enter into the filtrate. However, small things like water, electrolytes, glucose, amino acids, lipids, vitamins, and waste products do. And that's what we want. That's what we want. So all that stuff then is filtered out and is captured then by this catch basin, this Bowman's capsule, which is also simple squamous epithelium. And then all that then is directed into that proximal convoluted tubule, and here's the beginnings of that. Now I mentioned that hydrostatic pressure. As the blood flows into this afferent arteriole, it is suddenly constricted to a small diameter. That creates a high pressure back here, and as that blood keeps pushing in, that increases the pressure, and that's what forces substances out. That's where that hydrostatic pressure comes from. Now, this and we can change the filtration rate. The nephron in the body and nervous system and other hormones can change the filtration rate. So we're getting more than 125 liters a minute or less, depending on what we're doing. There's several mechanisms that do allow changes in that filtration. I'm just going to mention them right now, since we're talking about filtration. We have something called autoregulation, specifically what's called renal autoregulation. This refers to the nephron's ability, or the ability of the cells of the nephron, to regulate the filtration rate on its own. There's local factors. In other words, chemicals secreted from cells within the nephron that can work to change the filtration rate, either increase or decrease it. If you increase the filtration rate, you're going to get more filtrate. If you decrease the filtration rate, you get less filtrate. And that filtration rate basically is governed by hydrostatic pressure, which I won't get into all those dynamics. But renal autoregulation is one of those ways. Then we have nervous system, specifically the autonomic nervous system. And that would be the sympathetic branch. What we see over here on this afferent arteriole is that there are smooth muscle cells that surround that afferent arteriole just as it becomes the glomerulus. And we see that sympathetic nerve fibers will innervate those cells. So norepinephrine and the amount of nerve signals that are releasing norepinephrine can help to regulate the filtration. These smooth muscle cells can contract and relax, causing constriction or dilation of this afferent arteriole, thereby increasing or decreasing the rate of filtration. So if you're exercising, we're going to decrease that filtration rate because we want more of that blood to be directed to the skeletal muscles and to the heart. Doesn't stop, it's just decreased. And then we also have what's called and we talked about this before, the renin-angiotensin system, which plays a role in filtration regulation. And I'm going to address that separately in a little more detail, or really review it with you, because we talked about it before in chapter 20. So this is how filtration is regulated. Your book goes into a lot of details about it. Goes into a lot of details about certain substances and how you can, in a lab or clinically, measure filtration rate, because there's some substances that we know are filtered out. And if we know how much is there and we know how much is captured in urine, we can really determine filtration rate. All right, anything else I want to mention? All right, let's look at